What is SCNT and how does it make human stem cell lines? Imagine being able to create a tiny version of a person's cells that can turn into any kind of tissue in the body. That's what scientists do with a process called somatic cell nuclear transfer, or SCNT. It's a method that could help develop new treatments for diseases and even repair damaged organs. The process starts with a special egg cell, called an oocyte, which normally would develop into a baby if fertilized. But in SCNT, scientists remove the egg's original nucleus, which contains its genetic material. This step is called enucleation. Then they take a somatic cell from a donor, like a skin cell, and extract its nucleus. This nucleus holds all the genetic information of that person. Next, they insert or fuse this somatic cell nucleus into the enucleated egg. This reconstructed egg is then stimulated with a small electrical pulse or chemical signals to trick it into thinking it has been fertilized. This activation causes the egg to start dividing and developing into an embryo. The cytoplasm of the egg contains factors that reprogram the somatic cell nucleus, resetting its gene activity to an embryonic state. As the embryo grows in the lab, it reaches the blastocyst stage, which is a tiny ball of about 100 cells. Inside this blastocyst, there is a group of cells called the inner cell mass. Scientists can carefully remove these cells and grow them into stem cell lines. These stem cells are genetically identical to the person who donated the original somatic cell. Unlike reproductive cloning, where the goal is to create a full organism, therapeutic cloning uses the embryo only to harvest stem cells. These stem cells can then be turned into different types of cells, like insulin-producing cells for diabetes or heart cells for heart disease. Because they match the donor's DNA, they reduce the risk of rejection if used for transplants. This technique raises important ethical questions, especially about the status of the embryo and the rights of the donor. Many believe that creating and destroying embryos for research should be carefully regulated. Others focus on the potential health benefits, like personalized medicine and tissue regeneration. In summary, somatic cell nuclear transfer is a way to produce stem cell lines by transferring a donor's nucleus into an enucleated egg. It reprograms the nucleus to an embryonic state, allowing scientists to grow stem cells that can help treat diseases. But it's essential to consider the ethical principles involved, like respect for life, informed consent, and fair access to these advanced medical technologies.